Hello, in today's video we are going to see how to use BLAST from NCBI. So the purpose of this is to identify, validate and annotate genes. So let me show you how. So open a new tab and go to NCBI, the National Center for Biotechnology Information, and go to BLAST. The purpose of this is to help you identify similar genes. Think of nucleotide BLAST as the Google for DNA sequences. So you need a sample FASTA file. So you can search for a FASTA file by simply saying sample FASTA file and going to the first link, which is this. So this you can see it's Homo sapiens, it's already labeled, but just for the sake of this project, let's Control A, Control C and paste it. So we paste this FASTA sequence in this query sequence table. So then you want to see what database you want to see it from. So I'm gonna go with the genomic survey sequences and then I just click BLAST. So what this does is it searches the entire database from DNA to search what is this closely related to. It helps you identify what this gene is from. So it is very helpful when it comes to PCR to identify what the gene is from, what organism it is from, right? So now you can see it is still searching, and in a few seconds it will show you the relevant genes that are closely related so that you can identify where this DNA sample is from. So now you can see it has loaded Homo sapiens, so which is correct because here also it's labeled as Homo sapiens, so we know that it's from the human gene. So this is a very simple way how to use BLAST from NCBI to identify and validate genes. Thank you for watching and follow for more.